Because of the microphone. If you want to move, people, the white hat people, is like nowadays they are like old, guiding him with the hip.
Which be the best one, yeah.
Like like 3 months ago or something.
Just
retire and
yeah. Hard stuff.
So
students it's more than it's more than just bio. The frequency is higher.
Okay, I see,
Yes, everything was a big dog.
I felt like the like other blog somebody the other day now you're like you want the white dog.
Yeah. But I keep changing my company that I already use. This one I buy, I love it so much. Like my dog. Cutie. Like this one, yeah, this was my dog, but she died. So I don't put it because I feel sad. She was really old. She died at 21 years.
Yeah, she lived very even for
Yes. And so it's so sad that I put my random dog to forget about it.
I take my phone. Okay. Sorry.
Yeah. I completely forgot. Okay, so let me introduce you our next speaker. So his name is Igor Schapiro. He studied chemistry and molecular materials at the University of Duisburg Essen in Germany. There he obtained his bachelor in 2004, and he obtained later his PhD at the same university under the supervision of Professor Volker Bass. Then he did 3 different postdoctoral stages. The first one with Massimo Gucci at the Berlin Green State University. Then the second one with Frank Miese at the Max Planck Institute for Chemical Energy Conversion in Mannheim. And finally with Stephen Hacker at the Institute of Physics and Chemistry of Materials in Strasbourg. In 2015, he became a senior lecturer at the Fritz Haber Center for Molecular Dynamic Research at the University of Jerusalem in Israel. And his research interests are focused in the study of chemical reactions in biological systems and also in the development of methods for studying them. So thank you again for having accepted our invitation, and yeah, the pleasure.
Okay, Ned, thank you very much for this kind introduction, and also thank you and Juanjo for the opportunity to present here in this school. So it's going to be the last topic of the school, and I'm actually going to build on the previous lecture about QMM and the combination with excited states. So the outline of my lecture is going to be the following. So I will first give you a quick introduction. I think many of the things I'm going to show you heard already. And then I'm going to focus on 2 topics. One is regarding the calculation of absorption spectra. and the other one is about exploring the excited state reactivity. Okay, so I think that yesterday in the lecture from Jesus, I think you have seen already a similar kind of introduction. So since I'm going to talk about excited state, I just wanted to to cover the base, and there's like a couple of different processes that happens when you deal with excited state or with photons. And so if you start in your ground state, and then the first thing that you do is you, of course, that needs to excite. And in this simple scheme, I'm only dealing with one excited state of the same spin multiplicity. So what can happen then is we can follow the potential energy surface and then decay to the ground state by radiating, emitting a photon. So this would be a fluorescence process. Then another possibility is that there is a non-radiative decay, which is passing through an avoided crossing, and this means that there is some energy gap, but it can be overcome without emitting a photon. And the third case, which is highly suitable for computer simulations, is the case where the reaction undergoes a non-radiative ultra-fast decay through a feature called conical intersection. And I think that also Jesus has covered this. So this allows a very fast decay from the excited state to the ground state. So now in my lecture, in the first part, I'm going to talk about spectra and why it is important and how to simulate them. And in the second part, I will cover how can we use simulation methods to understand non-radiative process, but also radiative process. Okay, so what are the available techniques? And of course, some of the things were already covered, so that's why my focus will be on the combination between QMMM, so it's multi-scale simulation, combined with excited states. Okay, so these are going to be the two topics, and also the exercise at the end is also going to cover these two topics. So, I mean, there have been already several lectures and a lot of equations, but I just wanted to tell you, why are we doing this? What's the motivation? So what's the point of calculating spectra and also study their activity? So one of the new exciting fields is called optogenetics. Have you heard about this new technique? No. Okay, I have a few slides on this. And so here we are developing molecules which can be used for in vivo experiments. So and I have follow-up slides on this. And then the other possible application is, for example, super resolution microscopy, which is based on fluorescent molecules. So here you see a regular microscopic image, and this is super resolution, and this is achieved by using molecular probes, which can quickly switch on and off fluorescence. So it's desirable to understand how to control the reactivity in molecules, because then we can use it for this kind of applications. Now, I want to spend a few slides talking about optogenetics. So in general, QMMM methods are applied to highly complex systems with complex environments. And I'm going to talk exclusively about proteins. Right? So proteins which absorb light, they're called photoreceptor proteins. And these proteins can be used. You see the proteins are coded by DNA, as you heard in the previous lecture, by Elise. So you have the gene which is coding a specific protein, and then you have a promoter which is selecting a specific cell. And then in optogenetics, you use the genes for a specific molecule, you put them in the virus, and then you infect the mouse or any organism with this virus. And what happens is that the virus is infecting the brain, and then this promoter is specialized on one specific neuron. And this specific neuron can be, for example, in charge for circadian rhythm. It can be for optical, I don't know, for some sensing of the environment. Or in many cases, this is not known a priori. And then people in neuroscience, they study, they infect different neurons, and then by using light, they switch neurons on and off. So this allows you to do the experiment in living organisms. Because previously in neuroscience, you just take out the brain from the mouse and then you slice it and you do experiment on this end. This cannot really tell you how it will affect the behavior. But now with this optogenetics, you can do the experiment on living organisms. And so what happens next is then in a very specific neuron in the brain, a light-sensitive molecule is expressed, and then with light, you can activate this molecule. So, however, since if we're dealing with an experiment which is, it's about brain activity or neural activity, we need to deliver somehow the light, right? So this is done by a fiber optic cable. So there is a little hole drilled in the skull, and then this fiber optic cable can illuminate neural cells. So this was a lot of theory, but this is like a real experiment. See, this is a mouse, and here is the fiber optic cable. So the mouse is quite peaceful, but then when the laser is switched on, it activates some hunting. It activates the hunting behavior, and then it starts to move and attack this object here. So it's just one example to demonstrate that now you can really do experiment on living organism, and it's a big advancement for neuroscience and other fields. Okay, the question is like, how can we as computational chemists contribute to this? It seems to be very far off from what you have heard so far. So the thing is that in order to do this optogenetic experiment, they had to drill a hole in the skull of the mouse, because the light would not penetrate deeply enough in order to activate the brain, right? However, there is if you look at the absorption spectrum in tissue, there are the major components which absorb light in this region, in the visible, are hemoglobin and hemoglobin, some other modifications. So you see, this is an absorption spectrum of hemoglobin derivatives, and this intensity here, given by molar extinction coefficient, is on a logarithmic scale. So you see that the absorption is very high, but then there is this range from 650 to 900, where it's actually significantly lower. And after 900, the absorption of water that starts to kick in. So there is this sweet spot. It's called the optical transparency window. And in this window, you can actually illuminate. The light can go through skin. So this transparency window, and the experiment that I've shown you before with the mouse is done with a very specific molecule called channel rhodopsin 2. And so the absorption of this channel rhodopsin 2 is around 480 nanometers. So you see, it's outside of the transparency window, and that's why you need to drill a hole and to use an optical fiber cable. But if you manage to modify this channel rhodopsin and shift it to the red, then it's no longer necessary to drill a hole. Then you can do it non-invasively. And this is like one of the major questions in optogenetics: how to tune the absorption spectrum in order to reach this transparency window. And just to illustrate this concept, I'm going to do now a live experiment. So you see, I have two lasers. One is green color and the other one is red. And just to show you the power of this transparency window, this is a green laser. I'm holding my finger, you see? Can you see the green light? No. Because green light is outside of the transparency window and doesn't penetrate the skin. Now I'm using a red laser. And you see the red light goes through the skin. So this is our goal. We want to computationally achieve this shift. We want to modify somehow this molecule and put the absorption maximum in this transparency window. So this is called spectral tuning, because we're tuning the spectral properties. And now there are two different strategies you can imagine for this type of optical probes, because they are composed of a chromophore and a protein. So one way to modify it is to change the chromophore, right? And here I'm showing you an example. This is the retinal before it's bound by the protein, and here are different modifications. So you see the changes in the chromophore are highlighted in different colors. So there is this modification by adding a longer, bigger aromatic ring, and then also you can change the position of the methyl groups, and then by this affect the interaction with the protein. This picture is taken from Megan. Megan is also here in the audience, and she studied the effect of different modifications. Now, the other strategy is instead of changing the chromophore, you change the environment. So in general, if you do wet lab chemistry, you can change the solvent, and then you have this called phytochromism. So you can have a polar solvent, you can have charged, non-charged, non-polar, and this will affect the chromophore. But now today I'm going to talk exclusively about proteins. So our solvent is basically the protein environment. And now, how does it look? So if you look inside the protein in the binding pocket, you see this is the retinal, which is bound covalently to a lysine. It is surrounded by different residues. And these residues can have very different properties. Here, for example, you see tryptophan, which is a very large side chain. It's aromatic and non-charged. But then you have also carboxylates, so they can be deprotonated, and then they have a negative charge. But then you have also, I think this looks like alanine, so it's a neutral residue without any charge. So you can really modify the environment. You can introduce aromatic side chains. You can make it polar, non-polar, sterically large, small, and charged, positive or negative. So there is a lot of options to modify the environment. And now, before I move on to the next slide, I wanted to ask, which one do you think is more suitable for biotechnological applications, to modify the chromophore or the environment? Okay, let's say who thinks chromophore modification of the chromophore is better? Please raise your hand. And who thinks the environment? Okay. Why do you think the environment is better?
Because it has maybe more flexibility. So
okay, I mean, I would argue that for biotechnological applications, because you want to do many experiments, right? If you want to modify the chromophore, then you need a synthetic chemist who will be in the lab and synthesizing this molecule. But for protein, you don't actually need to work in the lab. You can let the bacteria work for you. So how it works? You have some gene that you want to that is coding a protein. You insert it in this DNA, and then you transfer it in the cell or transfect the cell. And then this particular cell, typically is E. coli, will grow and it will produce the protein. So at the end, you just destroy the cell, and by protein purification, you extract the protein that you want to do. So you don't need to do any work in the lab. You just once transfect the cell, and then the cell will produce the protein for you. So this is much more convenient because you don't need to actually buy this compound or synthesize it all. So this is like one of the major advantages of using proteins. So and yeah.
Can you actually tune so much in Redshift
Yes, I have the answer in the next slide.
Yeah, with the environment, let's say with the modification.
Yes. So there's like one common example for this tuning is basically the color vision in humans. So we have, this is the human eye, and you see the backside has the retina, this orange layer, and the retina has two types of cells. One is the elongated shape, it's called the rod cell, and the other one is called the cone cell. So inside, in the rod cell, we have a pigment which absorbs at 500 nanometers. So this is this dashed line spectrum, and there's only one pigment. But in the cone cells, which we actually have much fewer of them in our eye, there are three types of cells. They absorb short, medium, and long wavelengths, and this translates to blue, green, and red. So all of this absorption is achieved by a protein called opsin, and they have the same chromophore, just the environment is changing. Okay, so it's possible to tune it from 420 to 564, and I'm going to show it to you in a few slides that we can now go beyond 600. We can reach even 700 just by changing the protein. Okay, so now I mentioned it already a few times. So most of my research and also the slides are kind of focused on the rhodopsins. So what are the rhodopsins? Maybe not all of you are familiar with it. So there's two different types. You can distinguish between microbial rhodopsins, which are called type 1, which are found in lower organisms, and type 2 animal rhodopsins. So they share the same architecture. They have 7 transmembrane helices, and they are arranged kind of like a barrel. And in the middle of the barrel, you have the chromophore. So the chromophore is very well shielded from the environment. And these helices, transmembrane helices, they are embedded in the membrane. So this is like a thing that you cannot, this is very difficult to achieve with an organic molecule, because the membrane of the cell isolates the inside from the outside. So it allows you to regulate what will go inside, what will go outside. And this is indeed what is exploited in optogenetics. Because by using light, you can conduct ions in and out of the cell. This is what triggers the neurons. But okay, this was maybe too much information. So in type 1, you have basically the initial process is the all trans to certain cis isomerization. But in animal rhodopsins, it's present in 11 cis and goes to all trans. And then many microbial rhodopsins function as ion pumps or channels. This is the case of channel rhodopsin. Or now there is a new family of enzyme rhodopsins. And they undergo a photocycle. So you excite them, they undergo a photoisomerization, and then there is a change of protein conformation in many steps. And at the end, you just gain again, at the end of the photocycle, you gain the all trans retinal, and you can start the process again. Now, in animal rhodopsins, which are usually functioning as for visual and non-visual phototransduction, we don't have a cycle. So you excite it, it goes to all trans. There is a series of conformational changes, but at the end of the process, the retinal is cleaved from the protein and it exits the protein. So you can do this experiment only once. So there is an additional enzyme which re-isomerizes it and puts it back in rhodopsin. Anyway, so introduced to you rhodopsin, but how does it work? How can we tune the absorption of retinal? So actually, it goes back already to 1975. See, this was the time where theoreticians could still publish in Nature, and so they studied a model of the retinal, and they found that actually there is. I mean, they were focused on the isomerization, but they found that the positive charge on the shift base is migrating when you excite it. And then 3 years later, Aryeh Warshel, he published this paper. This is the 11 cis retinal, and you see this is the ground state charge distribution, and this is the excited state. So he found that the charge is moving from one end of the molecule to the other end in the excited state. And this rearrangement of the charge causes the bonds to rearrange. So this was this first starting point, and then the breakthrough was in 1979, so before all of you were even born. There was the group from Barry Honig and Keiichi Nakanishi. They proposed something called the external point charge model. And you need to imagine this was in 1979, so before you had personal computers, before any supercomputer centers, and before even software was written. So they had to really design their own packages and do the simulations. So, and just a funny fact, Barry Honig at the time was also at the Hebrew University where I am now. But we didn't overlap. So they proposed the following mechanism. So you have the charge distribution of the retinal in the ground state. The positive charge is here. This is called the shift base. It's the double bond between the carbon and nitrogen. And then when you excite it, the positive charge moves from the shift base to the beta ion ring. So now you have the distribution in the ground state and in the excited state, and it's spatially different. So how can we play with it? Okay, first of all, this was some schematic representation from the 70s, but today we can really calculate the charge distribution, and you can see there is more blue here compared to the excited state, where the blue is moving to the ring. So, and then each charge distribution is associated with a different electronic state, and the gap between them is corresponding to an excitation. So how can we tune the absorption? Now let's imagine there is a charge residue. Okay, this is a carboxylate. It could be a glutamate or aspartate residue. It's negatively charged and it's close to the shift base. So they form something called salt bridge. Salt bridge is a combination of ionic interaction. So we have positive and negative charge, and also a hydrogen bond. So, and then what happens is that this interaction in the ground state is much stronger than in the excited state, because the positive charge is moving from here to here. So in the excited state, there is just less positive charge. And this causes a differential effect. Okay? So now if you introduce a charge, you see the ground state is stabilized more because there is more positive charge, and the excited state is stabilized less, if stabilized at all. And so this causes the excitation energy to change. And this will go to higher excitation energy, which is causing a blue shift. Now you can play the game and put the charge on the other side. And now in the excited state, there is more positive charge. So it means this charge is going to stabilize the excited state more than the ground state, because the negative charge is close to the positive. And this will cause the excitation energy to get smaller, and in turn, it will cause a redshift. Okay? So this was the idea between the point charge model of Nakanishi and Honig. And the thing is that they did it in 1979. So at that time, there was no crystal structure available. So they didn't know exactly where the amino acids placed. And it turned out almost 20 years later, when the first structure of bacterial rhodopsin was solved, that indeed there is a counter-ion in this location. But there is no counter-ion in this location. So as I said, this was just theoretical work. I mean, but 5 years ago, the group of Lars Andersen from Denmark and Joni Tocker, together with us, we studied experimentally the absorption of the retinal in the gas phase to prove that really the counter-ion or the point charge has a strong effect. And so here you see two different retinals were studied, and then to the retinal they added the counter-ion in the gas phase. And so this was like a Twitter ion because it has positive and negative end. And indeed, you see these dots are the experimental spectra, and the sticks are our computations. So when you go from the pure retinal in blue and you add the beta-in counter-ion, you absorb a strong blue shift. And then you have a modified retinal, you add the counter-ion, you also have a blue shift. So this proves indeed that these considerations from the 70s were correct. So, but nowadays we have supercomputers, we have the crystal structure, and we have very accurate meshes. So can we do better? Here's an example of the current research. So here is Proteorhodopsin, and Proteorhodopsin is found in the ocean. And some of these bacteria which have this rhodopsin, they're distributed at different depths. If they're close to the surface, they would absorb more blue light, but if they're deeper, they absorb more green light. The bacteria have adapted to this situation, and they went through evolution. And there is one mutation in the position 105. In the blue Proteorhodopsin, there is a glutamine in this position. But in the green Proteorhodopsin, there's a leucine. So this is the polar residue, and this is a neutral one. And this is very far from the two poles, right? From the beta-ion ring being here and the shift base. So it's not really clear how it was tuned. However, you can clearly see this is a map where the charges from the environment were projected onto the chromophore. And you can clearly see this strong red color. This is caused by these two counter-ions, D97 and D227. So we could, when we did the simulations, we couldn't really see the effect of this position 105. So then what we did, we switched off all the charges because the counter-ion was so overwhelming, the effect. And we just projected the charge of the position 105. And then we see indeed the polar residue was causing quite a large shift. Polarization, but the neutral one was not polarizing the retinal. So, and then we looked, how does the electron distribution change between the excited and the ground state? And what we saw is, you see the difference between the excited and the ground state. So I think positive was red color, negative was blue color. So you see, indeed, there is a hot spot very close to this position 105, where the change of the excited and the ground state density is the biggest, the largest. And so this is exactly the position where this glutamine is polarizing the retinal. So this is the reason, and we were able to explain this interesting spectral tuning for the rhodopsin using QMMM simulations. Okay, now coming back to Vito's question. So you asked about can we really shift into the red? So I have to say that like during the years, in the last maybe 10, 15 years, there was a large attempt to design it computationally, but no one ever managed to shift the absorption beyond 600. However, very recently there was a modified channel rhodopsin which absorbs at 610. But just 2 years ago, there was a discovery of a new bestrophin rhodopsin fusion, which was absorbing at 661. So this is already in the transparency window. And the question was like, what's the mechanism behind it? So the experimentalists found this protein, they measured the absorption spectrum, they solved the structure, but it was still not clear what's the tuning mechanism. And this is where we stepped in with our QMMM simulations. And we looked, we have done the simulation, and what we found is very interesting. This is the structure of the binding pocket. You see in green the retinal. And what's interesting is that usually in rhodopsins, there's like 1 or 2 counterions. But in this case, there's 3 counterions. So 1, 2, and 3. And counterions means that this shift-based bond is positively charged, and there is an opposite negative charge around. And this was very unusual. So, and then in addition to that, there was a pH titration and measurement of the spectrum from a group in Japan. And they found that actually there are 3 different absorption maxima. 661 corresponds to pH value of 7, but if you increase the pH, the absorption goes to the blue. And then if you go to extremely high pH value, it goes to 362. The value of 377 is due to free retinal, which is not inside the protein. So then we did our QMMM simulation, and we found out that actually different protonation states of the 3 counterions are responsible for the shift. So if you protonate 3 of the 2 if you protonate 2 of the 3 counterions, you get the biggest shift. If you have only 1 of 3 protonated, you get a blue shift. And if you have none of the counterions protonated, what happens is that this counterion takes the proton from the shift base, and then the shift base becomes deprotonated. So here you see the shift base is protonated, here it's protonated, but then during the simulation, the proton was extracted. So, and this causes the biggest shift to the blue. So we were able to, using QMMM simulations, we were able to understand. And now, based on our investigation, now there are experimental groups who try to introduce by mutation, they try to introduce the 3 counterions in other rhodopsins and see if it really can shift to the red. Okay, so now I want to show you how we generate the spectrum. But I'm also going to introduce a new protein. And so I talked a lot about rhodopsins. Now I want to introduce a new exciting subfamily of phytochromes. So these are proteins, they're called cyanobacterial, and they belong to the superfamily of phytochromes. Phytochromes are photoreceptor in plants. They regulate the growth of plants and how they react to sunlight. So since you don't know about phytochromes, this is like a prototypical phytochrome, CPH1. It consists of 3 domains. This is called the phi domain, GAF domain, and the PAS domain. And the chromophore is found in the GAF domain. It's here in green. So this is a very large protein. However, cyanobacterial chromes have the same GAF domain as in phytochromes, but they are missing the PAS and phi domains. So they're very compact. The size of them is only 20 kilodaltons. So they can be really employed as optical probes or as tags for proteins. You can just attach them to proteins and express. So and the chromophore is not retinal, but the linear tetrapyrrole. So you're probably familiar with hemoglobin or what's it called, the chlorophylls. So they are also composed of 4 pyrrole rings, but they are usually closed, and there is a metal inside. The linear tetrapyrrole is a metabolic product from chlorophylls. So you see the chemical bond between the A and D ring is broken. So that's why it's called linear. We have 4 rings attached to each other, and they are covalently linked to a cysteine in the protein. So there is a chemical bond which keeps the chromophore in the protein. So now the cool thing about cyanobacterial chromes is that actually there are many types in nature and they cover the whole spectrum. While the phytochromes, the classical photoreceptor in plants, they only absorb in the red. So there is this question, what makes this cyanobacterial chrome so flexible? Why do they cover the whole spectrum? And what's the origin of the spectral tuning mechanism? Okay, and in addition to this is more linked to the second topic. In some of the cyanobacterial chromes, there is a considerable fluorescence noted, while in the phytochrome, there is no fluorescence. Okay, so we have worked on the specific cyanobacterial chrome, which is present in Synechocystis PCC6803. This is, for those who see it for the first time, it looks quite complicated, but this is the working horse in photosynthesis. So this is like the hydrogen molecule for quantum chemistry. So all the photosynthetic research is done with these cells. And in these cells, there is a protein called SLR1393G3. It has 156 amino acids, and it's a cyanobacterial chrome. And the interesting thing is that it absorbs 650 nanometers. Okay, so it could be well suited for optogenetics. But then, when you excite it, the red state is undergoing a photoisomerization. So one of the rings is flipping and causes the product to absorb in the red, in the green. Sorry. So this is the isomerized product, and then now the new spectrum is peaked at 536 nanometers. So and this is in total contrast to all the other phytochromes, because typical phytochrome would also absorb in the red. See, this is the phytochrome CPH1 that I showed you before. The dark adapted spectrum is at 600 something. And then when you excite it, the product is absorbing in the far red. So here the tuning is in the opposite direction. Here, upon isomerization, it goes to far red, and here it comes to tunes to the green. So there is a difference of almost 200 nanometers. So the question is, what causes this different shift, and how does the protein tune the absorption? So, and of course, we are not the first one to ask this question. Indeed, there were a lot of experiments already carried out. So, and there were, when we started the project, there were 4 different hypotheses. One of them was claiming that during the conversion from the red to the green form, the phycocyanobilin chromophore converts to phycobiolobilin. And what it means is simply there's one double bond is getting saturated. So the conjugation is much shorter, and this would explain the green shift. The other mechanism was based on protochromism. And here it was proposed that the C pyrrole ring is getting deprotonated in this position, and this causes the shift to the green. And then another idea was that this D ring, when it isomerizes, it removes one of the big side chains, and then water can enter in the binding pocket, and this water is causing the shift. And last but not least, the 4th suggestion was that during the rotation, the ring is trapped. It cannot fully rotate. It's trapped by the amino acid side chains. This is called the trap twist mechanism. So while we were working on it, 2 of the mechanisms were ruled out by resonance Raman spectroscopy and NMR, and they could clearly see that it's protonated in all the forms, and they could see that all the double bonds are intact. So there are 2 mechanisms which remain, and this was the starting point for our investigation. Okay, so now I wanted to say, in few words, I wanted to explain why is this protein not studied before? Why? I mean, it seems like a very promising candidate for biotechnological applications. The thing is that the first reason why it was not studied before is because of the large size of the chromophores. You see the chromophore PYP. This is from GFP. This is tryptophan, flavin. My favorite molecule, retinal. And you see the tetrapyrrole is twice the size of retinal. So if you want to do very accurate simulations, you need to treat this huge chromophore. And the other more important reason is there was no crystal structure. So we need, in order to do this QMM simulations, and you will see it later in the exercise, we need a crystal structure as a starting point. And this was not available for both forms, for the red and for the green. So far, there was only the red form from one type, and the green form was from a different type of protein. And also there was a blue form. But we need really both forms, the red and the green, from the same protein. Otherwise, we won't be able to make the comparison. So, and lucky for us, the group of Wolfgang Gärtner solved the crystal structure for the red form and for the green form, and we were able to do our simulations. So now I'm going to talk more about the methodology. I think you heard already a whole lecture on QMM by Elise. So I think I can skip over this. This is just the three gentlemen who got a Nobel Prize for development of multi-scale methods. And just to sum it up a lecture of 2 hours in one slide. So this you see is the photograph of Alfred Nobel, and it's divided in 3 different zones. And these are zones of different resolution. Did you also show this slide? Okay. Okay, thanks. Okay, so you see different zones. So the face has the highest resolution because this is the characteristic of a person you can recognize. And so then if we go outside of this high resolution zone, you see the picture is pixelated, but it's still sufficient to recognize that he was wearing a black suit and a white shirt. And then if you go to the third zone, then everything is just blurry and you don't have any information. Now the same concept can be transferred to an enzyme or protein photoreceptor. You have an active site because the entire protein is not involved in the reaction. Usually the reaction, like in chemistry, takes place in a very small space. Usually there is a bond that is formed or broken, or the chromophore is extended over a very small space. So then we can focus on one specific active site, and we have the highest resolution, like on the face, meaning that we have electrons and nuclei. This is the resolution. So then we can excite electrons, or we can break bonds. But then if you go outside of the active region, the protein does not really participate, in our case, in the excitation. So, I mean, there is usually in the immediate environment, there might be some effect, but it's unlikely that, something very far away will have an effect on the absorption. So that's why we reduce the resolution and we use a classical force field. Okay? And now electrons are no longer resolved. They are included in the parameters implicitly, but they are not particles in our simulation. And by this separation and reduction of the resolution, we can tackle very large systems. So now we can describe this very large protein in a few seconds, while the QM simulation will still take some time. And then if it's necessary, we can go even to lower resolution and then use a dielectric constant to model the solvent, for example. Then we don't have any resolution. So it's an implicit solvent model. We don't even have nuclei. We just use the dielectric constant, and that's it. So if you want to apply now this multi-scale method for the example that I gave you, the first thing you need to do is you need to know what part is going to be QM and which part is going to be MM. So basically, we need to select a partition. So since this was a new system and we didn't find in the literature much references, we decided to test 2 different sizes. So one QM region contains 66 atoms. This is the gray molecule. And then we added a larger region composed of 106 atoms. And this includes this green labeled thing. So these are 2 propionates, histidine, aspartate, and cysteine. Okay, so then we tested 2 different regions, and we decided to compute the spectra. And now I want to tell you this is why we use QMM and excited state together. So typically, if you would study an isolated molecule and you would like to investigate the absorption spectrum, what you do in computational methods is you localize the minimum. Okay, so it assumes that you can easily find the minimum in the ground state. And from the minimum geometry, you calculate a vertical excitation. This is called the vertical excitation approximation. So you just, for the same geometry, you calculate the excitation energy. So the problem is that in the protein, you don't have this one single minimum. So and in fact, first of all, it's very difficult to find a minimum. And second, there's not one, but there are many, and many of them are equivalent. So what you need to do instead is you need to try to capture them. So you need to do sampling and get many conformations and calculate the spectrum not from one geometry, but from many geometries. There are different strategies to do it. I'm going to show you the one that we applied in this particular study, and we're going to use it later in the exercise. So what we are doing is we are running a molecular dynamic simulation. We start first from classically, and then we switch to QMMM trajectory. And from the QMM trajectory in the ground state, we take snapshots, and we calculate for each snapshot the excitation energy. So here is the protocol in detail. After a classical equilibration, we make a production run using DFTB, which is a semi-empirical method, and it allows us to run a simulation of 1 nanosecond. And then we combine it with Embr force field for the protein. And then, as I mentioned, we use 2 different regions. We get 100 snapshots every 10 picoseconds. And then we use, for the electronic structure, for the Q part, we use ADC2. And we compute 10 excited states for the small region and 15 for the large region. And then, of course, the interaction between the protein environment and the chromophore is taken into account by electrostatic embedding, as you heard in the previous lecture. Okay, so this is what we get. From 100 snapshots, we get 100 sticks for each electronic state. So you see, this is the first excited state in black. These are 100 sticks. And then you see the next state, there is a gap here. The next state is S2. And then the excitation energy is much more blue shifted, and the oscillator strength, the height of the peaks is also much smaller. So the brightest state is clearly this one here. And then there's, as we go higher in energy, there's more and more and more excited states. So now what we do is we put a small Gaussian for each stick, and we add all these Gaussians together, and then we get this absorption spectrum. So if you repeat the procedure for the red form and for the green form, then we get this following computed spectra. This is the red form here, this is the green form. The solid line is showing the small QM region, the dashed line is showing the large QM region, and the sticks are representing the experimental values. So what you see here is that we have reproduced both the shift. So you see from the red you go to the green, we get the shift, but also the relative change in the intensity. The red one has a higher absorption and the green one has a lower. And we got it correctly with the simulation. However, it's not quantitatively, so the experimental shift is 0.35 electron volts. We are getting with the small QM region 0.23 and with the large 0.25. So they're very similar, and that's why we decided to stick with the smaller QM region for more calculations. So what we did next is something that you cannot do experimentally. So we just take the protein structures from the snapshots, and we set the charges to 0 from the protein. So now the chromophore is still in the distorted conformation, but the charges of the environment are 0. So then we recompute the spectra, and we get this dark green and dark, let's say, brown spectra. Okay, and you see that there is a shift from the green to dark green, from red to brown, but the shift is almost constant because without the charges, the shift changes only by 0.04. Okay, so what does it mean? What can we learn from this? Well, it means that this mechanism of hydration is most likely not true, because we switched off all the charges of the protein and also of the water, and the shift did not change significantly. It's still to the green, and we captured most of it. So it's most likely not this hydration mechanism. So then we are left only with the trap twist, and this says that somehow the environment keeps the D ring, this last ring here, it keeps it in a twisted conformation. So then we went on, we measured the twist for 100 snapshots of the red and the green form. And you see here, this is the twist between the C and D ring, and this is the twist, sorry, this axis here is a twist between the A and B ring. So these are the two rings in the periphery. In the middle, the B and C ring, they don't twist much. So then we plotted each snapshot as a symbol here in this, and then we colored it according to the absorption. And you can clearly see, without any manipulation, we already see the formation of two clusters. And in the center of the cluster is the structure, so the absorption obtained. This is the experimental absorption maximum and the dihedral angle from the crystal structure. So you see it's kind of in the center of these clusters. So it indicates indeed something with the twisting of the rings could be related to the absorption maximum. So then we wanted to check for the correlation. And then we plotted, you see the twist of the A and B ring versus the red and the green absorption. And then the twist of the C and D ring versus the absorption of the red and green form. And then we computed the Pearson correlation coefficient. Do you know what's the Pearson correlation? So the Pearson correlation coefficient tells you if there is a correlation, as the name suggests, between two sets of data. If the coefficient is 0, I mean, it only tests for linear correlation. If it's 0, it means there is no correlation. If it's 1, it means it's perfectly correlated. So then we compared the angle between the A and B ring versus the excitation energy, and we found for the AB ring, there is no correlation at all, for both, for the red and for the green. But for the CD, we obtained for the red form a correlation coefficient of 0.1, and for the green form 0.4. So it looks like it's very, very little, almost negligible correlation. So it seems that it's not the twist itself alone, but maybe something else. And now we switch to wave function analysis using Theodore, which Felix introduced on the first day of the school. So what we did is we divided the chromophore in 3 different fragments. Okay, and then we plotted the electron hole correlation plot, or we constructed the electron hole correlation. So we are using Theodore, we are expressing the excitation as an electron hole event. Okay, so if in one excitation, in one snapshot, the hole and the electron are on the same fragment, we would have an entry on the diagonal. So for example, if it's on B and C fragment, we'll have an entry of 1. Now, if there is a charge transfer, if the hole is on BC and the electron is on D, then we'll have an entry on the off diagonal. So if we sum up these values for 100 snapshots, we'll get the following expression for the red form and for the green form. And then in the next step, we focus only on the off-diagonal elements because they describe charge transfer processes. So we sum up these 3 values and these 3 values, and then we call it the charge transfer number. And it seems that they correlate much better. So for the red form, the correlation coefficient increased from 0.1 to 0.5, and from 0.4 to 0.8. Okay? So it means this charge transfer number is a much better indicator, and it correlates much better. But what's the explanation for this? Well, the reason is that in the trap twist mechanism, you look only at the dihedral. But you know, this is not the only deformation. You can also have ring puckering. You can have stretch of the bond. The bond angle can be larger or smaller. These geometrical parameters, all of them contribute to shifts. And it seems that it's different between the green and the red form. Now, all this deformation will alter the conjugation. If you have some puckering or double bond is getting stretched, the conjugation is getting worse. And this will affect the spectral tuning. So by using wave function analysis, you don't need to find each individual contribution, but you can just look at the overall wave function and then capture all these effects together. So I'm now done with the first part, and I have here the summary. The take-home message is, for this specific example at the end, it looks like it was a trap twist mechanism rather than hydration. And then we found this charge transfer number, which you can obtain with Theodore, or we call it also effective conjugation length, is indeed in charge for the shift from red to green. And in more general terms, we establish a protocol. So you are going to, in the exercise, do this, follow this protocol, and calculate snapshots, and then get the spectrum. And this allows us to simulate spectra in very complex environments. And also, QMMM simulations can provide mechanistic insights. I showed you at the very beginning, although there was an experimental spectrum for bestrophin rhodopsin and the crystal structure was solved, it was still not clear what causes the tuning. Thanks to QMMM simulation, experimentalists got a clue, and now they follow it and try to express similar rhodopsins. Okay, so this is the end of the first part. I think it's great time to have a break. And if you have questions, you can talk to me during the break, or you can ask a question now. Yes.
So collecting the analysis after the MD, do you do it randomly, or do you use like criteria? No, it's important to select something specific, or just random?
Yeah, so we're not choosing them randomly. We just select equidistant points. So it's every 10 picoseconds we select one.
Okay, it doesn't have to have a specific criteria for.
Yeah, no, we're not. We're trying not to have any bias. We did also test, we extended later on, we extended from 100 to 1,000, but the spectral shifts did not change.
Can I ask you something?
Yes.
In the simulation you showed you have this migration of the proton. This was like, you apply some enhanced sampling technique, or was a spontaneous?
I was spontaneous, yeah.
So you don't know, if maybe the barrier is very high, but the initial structure was the good one to induce the proton transfer, like a kind of artifact,
Yeah, but the thing is that in order to observe this, we had to include we had the retinal in the QM region and also the silicon trans in the water, so it was very large, and we couldn't run such a big QMM for biosimulation. So we decided to do a geometry optimization, and then it's very quickly. And it kind of makes sense because you have one positive charge and 3 negative charges. So by transferring the proton, you reduce the positive and the negative. So
yeah, sure.
From the electrostatic perspective, it makes a lot of sense.
Okay. Any other question? Okay, so then let's do the break.
Okay.
Yeah,
I mean,
4 months.
I think this is the mission. There are examples.
Which basically is creation of this partial.
Yes. Yeah, there can be also other effects.
Okay.
There are like small effects, like I showed in the example of proteolysis. There was in the middle, like a polar residue instead of a neutral. Polar instead of neutral. This causes polarization in this spot. Electron density was changed. Also, there are other examples closest to the ring. We remove some small then the ring rotates.
Okay. Yeah.
So the one of the downloads is then not connected.
So then you get dedicated to the green. Ya que se había quedado. Bueno, mira, casi que una no, eso debería enseñar en el colegio.
No, me explotó ¿cómo se llama? Okay, so now it's me between you and lunch. I promise to try to keep it short and easy to digest. So we're continuing with the second topic about the characterization of the excited field activity. So as I told you, there is this way some reactions pass through conical deflection, some of them get stuck in the excited state. And I will tell you now what are the different options that you can use to characterize different activities. So, of course, the most basic one is to do just simple geometry optimization and characterize stationary points. So you can optimize the ground state minimum, you can optimize the excited state minimum, and then you can also optimize crossing points. This is almost trivial. However, if you do it in a very complex environment, like in a protein, then it's very difficult to find and to be sure that you have found the absolute point. Another way to do it is to calculate a relaxed scan. So here you need to select one coordinate and then in different steps optimize it while relaxing all the other degrees of freedom. And then you can also use interpolation of coordinates. So you can connect this, for example, characterize geometric stationary points and then connect them by interpolation. Then there's also a way to do minimum energy pass, or if you do it in mass-weighted coordinates, it's called IRC. And then the best way to do it, if you have a very short lifetime in the excited state, so a reaction which goes through a conical intersection, it's quite convenient to do dynamics, and there are different flavors. So yesterday you heard from Jesus that there is this trajectory surface hopping, which is a kind of semi-classical way to do it. And then there are more accurate methods. Basil told you about ab initio multiple spawning, and I think Christina talked about quantum dynamics. So I'm not going to cover all of this, but I want to give you an example for each of these different techniques and discuss advantage and disadvantage. So let's start with the relaxed scan. So here is a very small model, like we have a truncated conjugated system, this truncated retinal. I'm showing only 5 double bonds, and we want to twist it around the central double bond. So we start by characterizing the ground state minimum, and then we get the Franconin point by calculating the excitation energy, and then in the next point we twist it in steps of 15 degrees. And then we collect points for both the excited and the ground state energy. And then at the end, we reach the conical intersection, and beyond, we are not able to get convergence if we optimize on the excited state. So the thing is that relaxed scan is convenient, and it should be like one of the first things to do when you have a new reaction, but it also comes with a disadvantage that it requires you to know the reaction coordinate. So in this simple case of a conjugated double bond system, it's quite intuitive that one double bond needs to twist. But in many cases, you don't know if it's an excited state proton transfer or if it's an isomerization. So sometimes relaxed scan is not the method of choice. And also, it suffers from convergence issues. So when you come close to degeneracy, sometimes it's hard to converge. And the other thing is also that while you constrain one or several specific coordinates, reaching from one point to another could actually rearrange the geometry significantly. And then it's very difficult to get the correct path. So an alternative to the relaxed scan, which is also computationally more efficient, is to do an interpolation. So this is done by first characterizing the reactant, and then again you compute the excitation and get the Frank-Condon point, and then instead of characterizing each single point, you can optimize the conical intersection if there is one, or if it's relevant for your system of interest, and then characterize the product. So you have now all the important points. You have the starting point, you have the intersection point, and you have the product. So in principle, you can just connect them with a straight line, which looks not very realistic for this isomerization. So what you do then is you calculate the interpolation in internal coordinates. So you express your molecular geometry in bonds, bond angles, and dihedrals, and you do it for different stationary points, and then you just make a linear interpolation. So when you then construct the surface from these interpolated geometries, usually you get a smooth pathway on the excited state, and then you can even connect the reactant, the product, and the conical intersection in the ground state and get also the relaxation. So this method is quite convenient. However, it requires that you optimize the conical intersection, which is not a trivial task. And I will show you later that this optimization of the conical intersection is not always realistic. And the other thing is that not all the paths occur to be linear. So if you imagine you have some bulky groups on the molecule, then at some point during the, for example, isomerization, these bulky groups need to pass each other. And this linear interpolation, you will just, they will go through. I mean, one molecular part will have to pass through another by some kind of tunneling. And this will increase the energy significantly. So it's not, it can be used, but it's not, it has also some disadvantage. So another way to study the reactivity is to calculate a minimum energy pathway. So you can typically a minimum energy pathway is run in the ground state, and then you start it from the transition state. But we have also adapted this minimum energy path to excited state processes. So here in this simple example, I'm showing you how to run the simulation. So you start as usual from the ground state geometry, excited to the Frank-Condon point, and then in the minimum energy path calculation, you define a hypersphere. Actually, you define the radius, and this is the collective coordinate for the whole system that you move. So now you have a well-defined radius of the hypersphere, and you look for the direction which will decrease the energy the most. So this process is called micro-iteration because you have a fixed starting, you have a fixed point, and you're searching in a specific hypersphere for the lowest point. So during this iterative process, you find the lowest, the biggest descent in energy, and then you define the new point. So moving from micro-iteration to macro-iteration is basically when you have the new point, and then you again form the hypersphere around the new geometry and move to the next point. So this just works out of the box. You can use any program package for minimum energy path calculation. You just need to specify that the gradient used for the minimum energy path is from the excited state. But then at some point, you're going to encounter a situation where the ground and excited state will be very close in energy. And then the program is getting confused. Then it's very difficult to converge because the energy gap between different electronic states is on the range of the energy that you change during the geometry optimization. So we have alleviated this problem by introducing surface hopping into minimum energy path calculation. So in the surface hopping algorithm that I used for this specific topic is that if there is a crossing inside the hypersphere, then the electronic structure is going to change between two consecutive steps. And this can be measured by looking at the change in the wave function. I think this might be also a topic that Felix has covered. So we are calculating the product between the wave function in the current step and the wave function in the next step. And if the product is deviating from orthogonality, if there is a significant change in the wave function, then we induce a hop, and the simulation is continued in the ground state. If you would not use the surface hopping, then the minimum energy pass calculation would stuck on the excited state, and you won't get any problems. But with this combination, we're able to characterize reactions which undergoes such a non-radiated decay. Then we can continue the minimum energy pass on the ground. Okay, so as all the methods I've shown before, there are of course also disadvantages. You can encounter barriers in the excited state, and with minimum energy pass calculation, you won't be able to overcome. So then you need to localize, you need to optimize the transition state, and restart the calculation from the transition state. And also, it doesn't really tell you about the branching of the product. So we can reach this point, but from a minimum energy calculation, you don't know like which portion of the population is going back to the reactant and which one is going to the product. This you can only obtain by running molecular dynamics. So molecular dynamics, as I mentioned in the previous slide, as you heard in this school, there are many flavors, and but in general, I think molecular dynamics, they are helpful to predict time-dependent properties like the reaction time or the rate. They can also help you to equilibrate structural geometry, and I would say it's the most realistic way to simulate a reaction if it's short enough. So what do we need for MD simulations? We need coordinates, velocities, and also forces. The forces can be obtained from the gradient. You just need to multiply it with -1. And in principle, you can use any possible method for MD. So I probably for this audience, I don't need to tell it, but if I'm talking to experimentalists or non-experts, when I say molecular dynamics, then everyone automatically assumes that I'm talking about classical molecular dynamics. But classical force field dynamics are not, this is just one way to do dynamics. You can put any force you want. So for this, I have a simple slide. We have this example potential energy surface. There is one point on the surface, and this surface can be computed using ab initio, quantum mechanical method. It can be a force field. It can be done using machine learning, whatever you want. You get the one starting point on the surface, and you assign the time t0 to it, and then you propagate in time, and you get the next point at t1, and then the next point at t2, and so on. So how do you make the propagation in bond-off nanodynamics? So actually, you can get any method you want to get the potential energy. Here's the quantum mechanical method. You solve Schrödinger equation, and then you get potential energy. And then you can form a derivative of the potential energy, and the negative of this derivative, it will be the force acting on each atom. And then from the force, you can get the acceleration. The force is just the mass time acceleration. The mass is known because you have the molecule, so you know the mass of each atom. And then you extract the acceleration, and then acceleration you can plug into Newton equation of motion, and using them and the time step information, you can get the change in velocity. So as you see here in this protocol, any method to generate the energy can be used. So in our case, we're using QMMM approach to get the QMMM energy and the gradient. Okay, I think this was already probably covered by Pedro in his lectures. So there are different ways to solve the Newton equation of motion. The Rayleigh family of algorithms, so here 3 types. This is classical textbook knowledge. So there is the original method from Rayleigh. This is the leapfrog method, and this is the velocity formulation. So important is to notice that we have 3 types of data: position, velocity, acceleration. And in order to, for molecular dynamics, it's useful to get the potential energy based on the coordinates and the velocities from the same time point, because then you can get the kinetic energy and potential energy for the same time. Okay, so since this was already covered, I will go very briefly over it. The most common way is the velocity Rayleigh algorithm. So here at a given time point t, you have all the information, the position, velocity, and acceleration, and you solve this equation here to get the position for the next point, the coordinates for the next point in time. And then you also, giving the information and the acceleration, you get the half-step velocity. This is done in the first step, but then in the second step, you take this new geometry and you calculate the force for the new geometry. And this is done in the energy and gradient calculation. And then you go to the second step, where you take the half-step velocities with the new forces from the time t plus delta t, and then you get the velocity for the full step, so t plus delta t. And this is the way how to propagate, and and then you can restart this cycle and again propagate to the next one. So, but the question is, so this is the equation, you've probably seen it many times. Why is it important to run molecular dynamics? So this is a very simple reaction. It's just in the ground state. And this is some peroxide together with a fluoride. So here you see a potential energy surface, and the point on top is the transition state. So now in this study by the group of Bill Hayes, they have done an IRC calculation, and this followed the red path. It reaches this deep minimum, and then they use exactly the same electronic structure method. They use, in this case, I think TFT. Okay, and they run a molecular dynamics simulation from precisely the same point. And you see the molecular dynamics simulation follows this black line. It doesn't go to the minimum. It escapes this deep minimum, and then it leads to dissociation of the complex. So now we have a three-body dissociation, three molecules are created. And in contrast, the geometry optimization leads to a complex of a D-alcohol with a fluoride. So you see the same electronic structure method, but different way of simulation gives completely different outcome. So apparently kinetic energy allows you to overcome small barriers and escape the minimum. And that's why it's really important already in the ground state. But as I'm showing you here, in this study by Todd Martinez, it has also, of course, an effect on excited state dynamics. There, because we have several surfaces, and then the kinetic energy becomes even more important. So in this study, a very simple molecule, ethylene, was studied using Ames. And what they found is that there is not only one conical intersection, which is characterized by a 90-degree twist, but there is another one where the proton from one carbon is migrating to the other carbon. So we have on one side one hydrogen, on the other side three. So this is what they discovered using dynamics. But they also discovered that the transition from the excited state to the ground state doesn't need to, to go exactly through a conical intersection where the energy gap is zero. The transition, and this is plotted here, the energy gap, you see the transition occurred at energy gaps which are much larger than zero. So they were transitioned at energies of 0.15 electrons. So you don't really need to go through a conical intersection. Already, the coupling is already strong in the vicinity. And you can have transitions much earlier. So the static way to characterize the reaction by plotting a conical intersection point and then making interpolation, this might be not the most accurate. And another evidence towards this is that the transition points where the hop occurred in this simulation, or okay, in Ames it should be called spawning. It was several electron volts above the minimum energy conical intersection. So when you optimize the conical intersection, typically you find the lowest point of the conical intersection space, and the transition occurred, you see, up to 4 electron volts above the conical intersection, above the minimum energy conical intersection. So it indicates that the characterization from static methods might not be the most accurate. So I mentioned many times conical intersection, but just for those who might not be familiar with the concept, I just want to emphasize. So you heard, I'm sure that many of you have heard about the transition state. This is a concept from thermodynamics. It's a stationary point, and it's characterized by having one transition vector. And this transition vector leads from the reactant A to the product B. Now, for the excited state, conical intersection is as widely distributed as a transition state. In fact, all the molecules, if they're bigger than 3 atoms and not symmetric, have at least one conical intersection. However, in contrast to transition state, they are not stationary points, they are singularities, because the derivative of the conical intersection is not defined. And then, instead of having one transition vector, they are characterized by 2 vectors, which form the branching plane. The branching plane is the plane which lifts the degeneracy between 2 electronic states. And conical intersections lead not only to one product, but they can lead to more than one product. So it's more complicated. So to illustrate this branching plane, so here is a simple molecule composed of 3 atoms, and there is one coordinate x1, the other coordinate x2, and the third coordinate alpha. So the branching plane is formed by x1 and x2 coordinates. You see that if you move along x1, which is a bond stretch, or x2 coordinate, which is another bond stretch, the degeneracy is lifted. But if you calculate the plane between alpha and x2 or alpha and x1, you see that the conical intersection becomes a C, because along the alpha coordinate you don't lift the degeneracy. Okay, so what is required in order to describe such degeneracies? Well, we have to rely on multi-configurational methods, and these multi-configurational methods, like the basic entrance level, like the most basic method is called CASSCF, which is short for complete active space self-consistent field method. So this method, the wave function, has 2 parameters. One is the CI expansion coefficient, and the other one is orbital coefficient. And then you need to define an active space, which is composed of orbitals and electrons. So how does it work? Well, in this method, you say, okay, I have some active orbitals which participate in the reaction. And so those which do not participate are called inactive. They're shown here in green. What does it mean, inactive? It means in the CASSCF wave function, the occupation is constantly 2. So the occupation in these orbitals does not change. But then we have an active space, which is defined by a number of orbitals and number of electrons. And here we allow all possible occupations. And then we have the third space, which is called the external or virtual space. In this space, the orbitals are always empty. So now in this active space, we allow electron substitutions. So we can go from one orbital to another, or we can promote even both of them. And then we make a linear combination. And the coefficients for different configurations are called the CI coefficients. So in this method, it's not only the orbitals that are optimized, but also the CI coefficient. And this allows for a very flexible wave function to describe degeneracies like the conical intersection. So later in the exercise, you will be able to calculate retinal, and then the active space here is based on the conjugated pi system. So we have 6 conjugated double bonds, and this results in 6 binding pi orbitals and 6 anti-binding orbitals. So the active space will be 12 electrons in 12 orbitals. The reason for this is because each double bond is defined by 2 p orbitals. 6 double bonds means 12 p orbitals, and then if they form a linear combination, 6 of them are bonding and 6 are anti-bonding. So what are the limitations of the methods? Well, the number of these possible configurations quickly increases factorial with the size of the active space. So there's this wild formula, which is depending on the electrons, the number of the electrons, capital N, number of molecular orbitals, and also the spin quantum number. You see this is 2 binomials. So if you calculate it for symmetric active space, you'll see that from a small active space of 2, 2, it really quickly increases from 3 to several hundred thousands. So this method is required to describe the conical intersection, but it's limited because due to the expansion of the active space, the calculation time is very, very long. Okay, so what about more efficient method computationally. What about TD-DFT? So there's another study by Todd Martinez group who showed that actually for TD-DFT, the topology around the conical intersection is not correctly described. As I showed you before, if you find the branching plane vectors, you would basically move out of the degeneracy along any of them. But for TD-DFT, it's not the case. You see here, this is CAS-PD2. This is done on top of CAS-CF. It's a perturbative correction. You see an intersection point, and around the point, the energy is increasing. But for TD-DFT, or I should say for linear response TD-DFT, this is not the case. Only in one dimension there is a degeneracy, which is lifted only, okay, it's lifted only in one dimension. In the other dimension, it's not lifted. So unfortunately, linear response TD-DFT, which is much more efficient, without any modification, is not suitable to describe. Okay, so this was about the theory for characterization of excited state reactivity. Now I want to show you one example. And this example is also related to the vision process I showed you before. So in the visual rhodopsins, the 11-cis retinal isomerizes to all trans. So what you see here is that this double bond, which is carbon 11 and carbon 12, is rotating from cis to trans upon light illumination. And this is one of the most, one of the best optimized reaction in nature because it has an excited state lifetime of less than 200 femtoseconds. Actually, the product is already observed after 200 in the ground state. And it's very efficient. 2/3 of the molecules go successfully from cis to trans. And it's only one single double bond that is selected. From these 6 double bonds, only one is isomerizing. Okay, and now this is in the protein environment. But if you take this molecule out and you put it in solution, then all these nice properties are lost. The lifetime is going from femtosecond to picosecond. The yield is going from 70% to below 20. And instead of one product, we have a distribution of many isomers. So somehow the protein environment is optimizing this reaction and allows it to happen in a very efficient way. So the question for this reaction is, how can the chromophore, this retinal, which is bound, it's linked through a chemical bond to the protein, how can it undergo a motion of 180 degrees? Because as I showed you in the first part of the lecture, the binding pocket is very tight. There is not enough space. If it would rotate, it would bump into the protein. But still, it's very efficient, and the reaction is completed in 200 femtoseconds. So there must be some interesting mechanisms. And one of the first theoretical studies was done in 1976 by Arye Warshel. He has used a computer from NASA to make this simulation. And he found out that actually 2 double bonds in the retinal isomerize at the same time. So 2 of them undergo this motion, and this motion resembles the motion of pedals in the bicycle. So he called it the bicycle pedal mechanism. The thing is, because one bond is rotating and the other one is moving as well, the space required for double bond isomerization is limited. Because in the classical, if you open an organic chemistry textbook, you see on the paper it looks like one end of the molecule is moving 180 degrees. This would require a lot of space. But if 2 double bonds are moving, then the space would be reduced. So this was really really pioneering work because there was no crystal structure, there were no computer programs. He wrote the program and he mimicked the environment of the protein by van der Waals views. Then a few years later, Robert Liu, who is based on Hawaii, he came up with the hula twist mechanism. So he was saying, probably inspired by local activities in Hawaii, he was saying that, instead of 2 double bonds, there is a double bond and a single bond next to it. They rotate at the same time. And he said that this hula twist mechanism would require even less volume inside the protein than the bicycle pedal. However, there was like one little detail that it was not leading to the all trans. It was leading to 11 trans, 10 assist retinal protein. So it was disagreeing with the experimental findings. But it didn't stop him from publishing many review articles on this topic. So then, a few years, few decades later, the Oribuchi group came up with the crankshaft mechanism. So it's basically an extension of the bicycle pedal where they say there is not only one double bond, but similar to a crankshaft, several double bonds twist a little bit, and overall it causes to reduction of the space required for the isomerization. And then the group from Hans Lischka came up with the folding table mechanism. Okay, so I gave this talk many times, but I still don't know where is the analogy between the table and the retinal. So they claim that the double bond in the middle is rotating by 90 degrees, but the two neighboring double bonds twist by 45. And by doing this folding table motion, they are reducing the space. And if you think this is funny, I have another one. So the group from Ishida came up with a wet towel motion, where actually, like a wet towel, the whole retinal is twisted along several bonds. In a way, it's very similar to the crankshaft. So we decided to also study it many years ago. And so in the first step, we have separated QM from MM. We decided the retinal to be part of the QM region, and we use this KCCF method here because we are dealing with the conical dissection. And then, like, the only thing was to introduce a link atom, because in case of retinal, it's covalently bound to the protein. So when we separate QM from MM, in solution it wouldn't be a problem because you take the retinal and the solvent molecules are separated. But in the protein, we need to cut it somewhere. And so we had to saturate this truncated bond with the link atom, and then we could do the simulation. And here, this is what you're going to do later in the exercise. So this is a flow chart of the QMM molecular dynamics with surface hopping. In Molkas, each different task is done by a different model, so it's generating integrals. This is from Nicolas Ferret for QMMM. Then this is the KCCF wave function, the gradients, and then the interaction part. And this is my implementation, surface hopping with molecular dynamics. So you can run everything now in one package. I think the professor showed yesterday Sharp, but here it's basically doing the same thing, but it's just integrated in one single package. So you don't need to switch the programs. So when you have a lot of time and you have this package, you can run many trajectories. I think back in the day when I was a postdoc, it was taking a few months. So we generated something like 20 or 30 trajectories. And here you see this is the ground state evolution. This is the excited state evolution. And so so I need to say that most of them, so these are different trajectories from different initial geometries and velocities. Most of them follow the same trend. So there is, in this region, around 100 femtosecond, there is a crossing. But you see also there are some cases where we have outliers. So one of these trajectories, for example, did not hop in the time of 200 femtoseconds. So this really emphasizes why we need, for such complicated environments, why we need to do sampling. And so now I selected one trajectory, and I'm going to go in detail over it because I want to show that with computational methods, we can really get insight that cannot be easily or cannot obtain at all from experiments. So here we have the ground state energy in black and the excited state energy in red. And the start of the simulation is at time point 0. Then around 100 femtoseconds, you see a dotted line, and this indicates a surface hop. This is the situation where the trajectory went through a transition from the excited state to the ground state. Now, if you calculate the difference between the red and the black curve, you see this is the blue curve, and the energy gap is quite large at the beginning, but then very quickly, after a few femtoseconds, it drops. And then it slowly oscillates and reaches almost 0, and then increases again. So if you're a physicist, I guess you're already satisfied with this result, but for chemists and for biologists, I think we want to know what's happening on the molecular level. So we're looking at the geometry. Here is the retinal, and this is the plot of the bond length. So in blue, you see. Okay, I don't have it here. So in blue, you see two single bonds. This is the 12-13 and 14-15. In red, you see 11-12 double bond and 13-14 double bond. If you look at time 0, you see this is how it should be. The double bond is short and the single bond is long. Right? But the moment we excite the molecule at time 0, you see something is happening. The single bond is getting contracted, it's getting shorter, and the double bond is getting stretched. So what we see is something called inversion of the bond length alternation. So the pattern of double, single, double, single, double is inverted to single, double, single, double. So this is happening on the excited state because the charge distribution has changed. The positive charge has moved from here to here. And the consequence is that while we are in the excited state, from here to here, the double bond is very long and the single bonds are all very short. Okay? Now, if we go back, you see the initial period where the energy dropped quite a lot coincides with this inversion. So in the first 10, 15, 20 seconds, the electrons are excited in time 0, but the nuclei are heavy, so it takes some time until the nuclei rearrange. And this is this very fast drop in the energy, and it is associated with the inversion of the bond length. But then, when we continue the simulation, the energy still drops, although the double bonds do not change. So this must be due to another reaction coordinate, and this is a dihedral. So I'm not sure what happened with my slides. I'll go back. So what happens here is once you return to the ground state, then the original bond order is restored with a little bit of delay. You see now the single bonds are again long and the double bonds are short. This is the scale of the length. But then the other coordinate is the dihedral angle, and as you know, the dihedral angle is defined by 4 atoms. The first 3 form a plane, and then the last 3 form a second plane, and the angle between the 2 planes is called the dihedral. So we have defined 2 different dihedral angles. The black line involves only carbons, so carbon 10, 11, 12, 13. And the red line is the same double bond, but with 2 hydrogens. So it's hydrogen, carbon, carbon, hydrogen. So it's described the same double bond, but of course, hydrogen can move much faster and react to any change much faster. And you see that the amplitude of the red curve is much higher than of the black because of the light hydrogen. But then we calculated the difference between them, which is called the Hooke mode. So this is the hydrogen out of plane mode, and it's the difference between the black and the red. So what you see here is that at the beginning, while the double bond is still very short, there is no much movement, but after the first 10, 15, 20 seconds, there is an onset of rotation. It goes up to 90 degrees at the time of hopping, and then in the ground state, it continues and forms the trans. So what I showed you with a few slides, I can also visualize in the movie. Here for clarity, I removed the protein. So the protein is present in the simulation, but I'm not showing it in the simulation. And the double bond, which is isomerizing, is the 11-12 double bond. It's highlighted in yellow. So here you see the geometrical changes of the retinal. You see that the twist of the double bond starts to increase. It goes through almost 90, and now we are back in the ground state, and the double bond is formed again. But now it's in a trans conformation. So this is the visualization of the geometry. But since it was part of the Cume region, we can also visualize the electron density. In this case, it's a subset of the density. I'm showing you only the pi electron density. only the pi orbitals and electrons are visualized. And you can already see that wherever you have the pi density, there you must have a double bond. And then in between, where there is not, these are single bonds. So I'm going to start the simulation now. And if you remember, the first coordinate is the inversion of the bond length alternation. So you'll see in a moment I'm going to press play. The double bond, the density is going to break. See this in the first few femtoseconds, the density broke here. So now the double bond can rotate. And now the hydrogen moves in plane and the other one moves out of the plane. Now this was 90 degrees, and now we are back in the ground state. You can see it because the density is again formed around the double bond. So now we have formed the trans product, and if you pay attention, I'm going to play the movie one more time. So the two ends of the retinal are not moving. This one is linked to the protein, and this one is also interacting with the side chain. But you see that both of them are trapped. The isomerization is not. It's not doing this big motion of 180 degrees, but instead the two ends are almost fixed. You see this end is in the same position and this also. So the reason for this is because the adjacent double bond, which is this one here and this one here, they also undergo some twist. In the case of the 9-10 double bond, there is a twist of 60 degrees. And in case of this 13-14, there is a smaller twist of 30 degrees. So because the two adjacent double bonds twist in opposite direction, the double bond in the middle can isomerize without a big conformational change. Okay, so now we studied the isomerization and we kind of found the mechanism. So we say that the double bond is indeed assisted by neighboring double bonds. It's very similar to the original idea from Ari Worsham, but it's not the full bicycle pedal because one double bond is twisted by 60 degrees and then it goes back. So we call it aborted bicycle pedal instead of introducing a new name. And so, okay, we studied the mechanism, but this is interesting, but like, how can we prove that our simulation indeed has some predictive power? Well, we looked at the geometry at the conical intersection. So this is called Newman projection. We're looking along the 11-12 double bond. And this is 90-degree twisted. So in front, we have this carbon 11 and a p orbital. And behind it is carbon 12 with another p orbital. And they are like in this orientation. So the overlap between p orbitals is basically minimum, because in order to have overlap, they need to be parallel or antiparallel. But here is 90 degrees. So at the conical intersection, you have the worst overlap between the orbitals. And then what happens is that after the conical intersection, the double bond is formed again, and now the motion of the hydrogens is really decisive. If these two hydrogen atoms move away from each other, then you see the lobes labeled B prime and B, they're going to overlap. And this will form the E isomer because the double bond is formed and now it cannot rotate anymore. But if they move towards each other, then A prime and B prime and A will overlap. And this will go to the back to the cis. So in nature, the ratio between trans and cis is roughly 65, 67 to 35. Okay, so 2/3 go successfully to trans, 1/3 goes back. So now, what's the role of this hoop mode? Because I said that the hydrogen moves out of the plane and this decides about the product. Well, if you remember, in this plot here, I showed you the hoop mode, that it undergoes this oscillation, and then it really decides about the product formation, because the hop, if it comes before or after hoop, will control if the double bond is formed or not. So the exact timing between the hoop mode oscillation, basically this motion here, and the hopping will decide about the outcome. So this is what we predicted throughout simulations. And then an experimental group tested this hypothesis. So what the group from Richard Matisse and from Berkeley and Kupura from Oxford, they synthesized or they measured this rhodopsin, and they replaced 2 hydrogen atoms in retinal by deuterium. So they had 3 different isotopomers. In one case, they replaced both hydrogen with deuterium, and in the other 2 cases, each of the hydrogen was replaced by 1 deuterium. So as you can imagine, in case of 2 deuterium substitutions, the Hooke mode was still symmetric, and it did not influence the ratio. The ratio remained the same. But in any of the isotopomers where only 1 hydrogen was replaced, now the Hooke mode is no longer symmetric, okay? And this really perturbed the timing between the Hooke and the pocket. So basically, they were able to revert. Now, the majority was going back to the cis, and the trans was going from 65 to 45%. So they indeed provided this experimental evidence for our prediction, and I think it was quite nice and a good motivation to move forward. So this is the second take-home message. So many methods can be used to study the activity in excited state, and you need to know, you need to try maybe several of them, and it also depends on which question you want to answer. And then QMMM, excited state dynamics, in my opinion, is the most realistic way to describe such light-induced processes. And also, as I showed in this example of rhodopsin, a really detailed mechanistic insight can be provided, which cannot be obtained experimentally. So I think without our simulations, they wouldn't have tried these deuterium isotopes. So I think it's really nice to see that experimentalists appreciate our simulations and also that the predictions are correct. And yeah, I think this is my take-home message, and with this I will conclude. And I just want to give you a preview of the practical session. So we're going to try to calculate spectra of a protein and use sampling for this purpose. And then in the second part, we'll do non-adiabatic dynamics and calculate the isomerization. So with this, I'm at the end. Thank you for your attention, and I'll be happy to take questions.
Okay, so time for questions.
Oh, we can also go for lunch.
Okay, so maybe you can ask questions during the computer exercise, but okay, so let's thank you, Igor. Thank you.